Dear viewers, Assalamu alaikum. Today we are going to discuss that how we can design pharmacophores or scaffolds for our compounds which are active against certain protein targets or macromolecules or enzymes. Let's suppose we have this compound which is benzene sulfonamide, and we can draw this type of compound or structure which is active and in ChEMDraw, and we have tutorials on this ChEMDraw. You can visit my videos. So here I have already made a structure. We can also make structure by right here convert name to structure. So I'm going to change this. So it is, let's suppose, hydroxybenzene sulfonamide. Okay, so this is our hydroxybenzene sulfonamide. Okay, we can make our structure, and so we can clear it. Okay, now we need, let's suppose we need a smiley structure for this compound, but in ChEMDraw we don't have an option that we can save this file in smileys format. We have some other options like JPG format, GIF, or mol file or SDF file, but we cannot export this file in smileys format. So for this we can copy this structure. So we can copy it by right clicking, copy, and we can paste it in Discovery Studio Visualizer. And for Discovery Studio Visualizer we have made a lot of tutorials on this. So here let's suppose I have pasted this structure. So here we can go to file, new molecule window, and then we will go to paste. Now this is our molecule which is benzene sulfonamide, and for this structure now we can save it in smileys format. So I am going to save this file in smileys. So we have a lot of options here. We can also save it here in image file, but I am going to XYZ coordinates file. I'm going to save it in smileys file on desktop. So I'm going to save it. So in this way we can get smileys file for our compound desired, which is required. So here we have smileys file. Like notepad, I'm going to open it. So this is our smiley file. I will select it and then copy this file. Okay, we can also go to PubChem and we can write here the name of our compound. If it is available in the PubChem library, then we get smileys file from here. Like here we have written here that is 3-nitrobenzene sulfonamide. So for this compound, here is our smileys file. This is our smileys file, or these are some other structures, but here we are only not going to similar structures. So in this way we get smileys file also from the library. And now I am going to make scaffolds. So we will use today we are going to discuss that we will use Swiss similarity or Swiss drug design. So this tool will be used today to make pharmacophores or scaffolds. So here let's suppose I have pasted here that here is our smileys, and now we can also build a structure here also by using this sketcher. Like here it is our smileys, so we can build here structure also by making this sketch. So this is our structure. The smileys of this structure are here, And after this we will select our class of compound, either it is synthesizable, commercial, bioactive, or already it is in drugs. So here we are not going to select, but we can here we will choose that which can be synthesizable. And then we will go to here, select compound library and screening method, and from here which are synthesizable, so we can go to scaffolds and we can select, like here these are different options which are available. But if we go to, like here, we look at that these are bioactive, then we have a lot of options. Like here we have ligand library present in PDB library, which are already bound with some enzyme or target protein, or we can use some other classes, like they are present in ChEMBL library with full database of 29. And GPCR class are some kinases or proteases. And from here we will select that either we are going to choose pharmacophore or only scaffold or generic scaffold. So we have these options available. Like here I am going to choose that I am going to make scaffold of ChEMBL full database. So after clicking here we will start screening. And if we are going to take that these compounds are synthesizable, then we can choose from here. Like here I am going to take Ottawa tangible, or we can use live chemical or some others, that ANOVA form or an amine. So from this okay, we have also option of 2D and 3D, and 2D and 3D combined, but here we don't have an option of 3D or 2D. But if we look at here, if we have the drug or bioactive or commercial, then we have an option of 3D also. These are electroshape or E3FP, so we can select from 3D structure also, that it should be 3D format with all the stereoisomers or some other kinds, so all are in option. So we are going to take, let's suppose, synthesizable, and this one, start screening. So here is our job of Swiss similarity search for Swiss drug design, and now it is going to calculate. Today is January 12, 2023. This shows the time, and estimated execution time is 117 seconds. Okay, we have also searched some, like here we have searched for some compounds, like here it is our nitro benzene sulfonamide compound. For this we have already searched ChEMBL library for our convenience, and we have a lot of similar compounds for this, and it is based on generic scaffold. And we have some others options, like here we have ChEMBL with scaffolds. And so here, okay, so when we have scaffolds like here, and I'm going to check that whether which compound it is. Let's suppose I'm going to check this compound, so we can go here, like it gives us report about this compound. So it will gives us compound report card, name and classification of the compound, how much research has been done on this compound, and other options. Molecular weight is 360, small molecule, and there is canonical smileys, Henkel key, and other literature is not available. Alternative forms are available, but the compound is not already searched, are not synthesized. So bioactivity summary is, these are the summaries, so we can check these. Like here we have another compound The report card of this compound tells us, so this compound, the data is not available for this compound, so it can be synthesized, so we can check this compound. So here we get our results for our Ottawa tongue scaffold, and these type of compounds are synthesizable, which are scaffolds of this compound, which is query molecule. We can retrieve data from here with different formats, and after this we can go to, from here we can check the ADME properties of this compound. Let's suppose I am interested with this compound, so I will go to target prediction of this, so how it will target or which targets it is going to hit, and also we can search similarity for this compound also. So we can search similarity for this compound also, that how much similar compounds are available for this compound. So these compounds are, it means not synthesized, and we can synthesize these compounds, but we can go to, let's suppose I am going to check the target prediction for this compound. So I am going to check target prediction. The job was submitted. After some time we will get the targets for this compound. So this is our query molecule, and the target classes are family of G protein coupled receptors is in blue, and kinases, which are 20%. So these are kinases which they hit kinases, 20% kinases, and this is 26.7%, which is lyases. So it is the most probable targets are lyases, and we can also get detail from here, like here it is going to hit family of G protein coupled receptors, which we have 2D, non-active 2Ds are 53, and serotonin, serine threonine protein kinase. So for kinase it has 13 3D structures which are non-active, which are similar to this one, and similarly kinases, phosphatases, and so, and the carbonic anhydrase 4 lyases. Two compounds are active. We can download here from here which are the non-active compounds. So non-actives can be retrieved from here. So here we have non-actives. So we can download the file from here, and let's suppose I am going to check here that it's Uniprot ID, ChEMBL ID, or this is common name. So we can go to search from here Uniprot ID for this compound. Similarly, we have searched Swiss target prediction for some other compounds, and I am going to show you that here, like here it is, it gives the probability. So this probability is most important, and this compound is going to hit G protein coupled receptors mostly, and scaffolds for this, if we have ligand present in the PDB, which is ligand expo. So for ligand expo we will get this type of information, like here these are the PDB entries, they are present in the PDB file format, like here it is for NL. So this is the ligand, which is aminophenol, and it is present here, and it will be a ligand with certain macromolecules. So we can search from here. So molecules from drug bank we can also search, like here from drug bank. If we are going to check from drug bank, we will get similar compounds like here. And the final thing I would like to show you, that if we are going to change this compound and we are going to add some fragment or remove some fragment, and we are going to check that either we can synthesize it or not. So we can get Smiley's file from here for this compound, and we get sent to Swiss bioisoesters. So this is Swiss bioisoesters, Swiss drug design, and this is our compound, and we can check that which search for possible replacement of a fragment. So we can check that how we can replace some fragments with some other. Let's suppose I am going to replace this fragment with some other fragments from here. Like here I am going to make this fragment, and I am going to replace it. So we will replace it by adding R. So from here it is R. Okay, now we are going to replace this R fragment with this one, and I am going to go to query database. Okay, so we did not. Okay, I am going to highlight this, that we are going to change some fragments like here. Look at here, I'm going to highlight this one, and now look at this, that can we change this? It is taking the whole fragment and it is going to change by this fragment, but so for this it is not possible. So I think it is taking fragments, so it is fragment 1 and fragment 2, but so we can draw a structure here, and like this we are going to okay, I am going to cut this one from here, or I am going to, let's suppose I am going to copy this structure. Copy, and I am going to delete this structure, erase this one. I'm going to cut this whole. So after this I am going to copy it and paste, or paste, Control V. We can use Control V for paste, and after that we can check that either this fragment can be copy, and so we have pasted here. Now I am going to cut this fragment. We are not interested, so I am going to cut this, and now I am going to change this fragment to this one. So it cannot be changed, so it tells us. And similarly we can check our fragments. So in this way we can make different scaffolds or pharmacophores, and we can select our desired compound. We can screen by ADME properties, or we can also screen our compound by docking with the enzyme, or we can further we can send it to Swiss Similarity to search similar compounds for this compound. And for this again we will go to here, and by selecting this we will go to the option that we are going to bioactive compounds, and then from this library and scaffolds, and then we will go to start screening. So in this way we will get similar compounds. So I hope you understand that how we can make different scaffolds or pharmacophores from our given active compounds. Okay, so thanks for watching my video. In our next video we will discuss that how we can make other pharmacophores or scaffolds by using some other tools. Thanks for watching my video.